So let's go back to our flower example for two reasons. One is flowers are easier to draw, and second, I like flowers. And so we've got our red flower and our pink flower. Now remember, these are, the gene is flower colour, the alleles are red or pink flower colour, and the reason for that is that the red flower makes a lot of a protein that appears red, so it makes a lot of that protein in its petals. The pink flower makes less. The reason for the variation was a mutation that changed the DNA. The DNA tells the cell, the petal cell, how much protein to make, in which cells, and how much. So the DNA tells our petals, make lots of red protein, make a little bit of red protein. Originally, they would have all been one colour. A mutation would have occurred that caused this variation, and the mutation persisted. It was spread throughout the population, and now we call them alleles. So we look at our flowers and we see, well, some of them are red and some of them are pink. And we've done a bit of study, and we've noticed that red flowers are dominant over pink flowers. That is, red flowers can have the genotype, big R, or big R, little r, and that's because dominant alleles turn up in your phenotype, your appearance, even if you've only got one of them. On the other hand, all the pink flowers must be little r. So red flowers can be homozygous dominant or heterozygous. Pink flowers can only be homozygous recessive. These two genotypes, big R and big R, little r, always lead to the phenotype of red, whereas this one always leads to the phenotype of pink. So how do we actually do this? And the answer is, well, we can predict something about it. Let's say we just get two red flowers, okay, and we breed them together. So I hope you can see that there's three different situations with the red flowers. The red flowers could both be homozygous dominant. And I hope you can see that if we had two homozygous dominant flowers, they would always give red flowers. Always, forever and ever and ever. Okay, as long as they're both homozygous dominant, there will never be any pink flowers. On the other hand, if one of our flowers was homozygous dominant but the other was heterozygous, we would still get, only get red flowers, but half of our red flowers would carry a recessive gene. And why that's important is in the third example, when you have two heterozygous flowers bred together. And when you have two heterozygous flowers bred together, mostly you get red flowers, but a quarter of your flowers will be pink. Now, this might not seem like a big deal, but gardeners are very particular about their flowers, and so many times they don't want this scenario. They don't want to breed together two flowers that are like expensive and blah blah blah, and then get these ugly pink ones that they don't like. So what they do is they want to guarantee that their flowers are pure breeding, their flowers are homozygous dominant. And to do that, they run something called a test cross. That is, they take a flower they are almost certain is homozygous dominant. And they're like, we're going to test you, little flower. We're going to be certain. And so they breed it with a pink flower. And the reason they choose a pink flower is they know that the pink flower is homozygous recessive. So they know that if they run that through, if they set up their test, if their flower actually is homozygous dominant, all the babies will be red, every single one. On the other hand, if their flower is actually homozygous, heterozygous, they know that half their babies will be pink. So they would, if this was your question in your exam, or if this was something you were doing because you're an enthusiastic gardener, you would say in this situation, 100% of the babies will have the heterozygous genotype and 100% of the babies will be red. On the other hand, if you do this, 50% of the babies will have the heterozygous genotype, but 50% will be homozygous recessive. Likewise, 50% of the babies will be red and 50% of the babies will be pink. Okay, there are a few things about this that you need to know. One is that there has to be plenty of babies. It's 50%, and each episode of fertilization, each time a seed is fertilized, a flower is fertilized, creating a seed, it's an independent event. It's not like if you've got 10 seeds, you're guaranteed that half them will be one way and half them will be the other. It's a random event. Every time fertilization occurs, it's its own separate event. It's randomly predicted. That's why you can have a family with, such wild variation in it, because you only have 3 or 4 children in a family, and so you might have a blue-eyed parent and a not blue-eyed parent, but all the babies have brown eyes, not blue eyes, because it's random every time. You need to have lots of babies to be certain. So that's one of the reasons why I chose flowers as well, because we can have 200 babies really easily. We just grow 200 seeds. Simple. Animals are more difficult because you don't really want to have 100 babies. You probably only want to have a few babies. So as long as there's enough offspring, you can be sure by this test of what the genotype of the parents are.